human existence, the events occurring in the reproductive tract of a female as its embryo undergoes the early stages of its growth have been hidden from us. With the development of methods to grow embryos in the laboratory and to see them through time-lapse microscopy, this hidden world is now revealed.
In the cow, the first 4 to 5 days of development occur in the oviduct, called fallopian tubes in humans. The embryo then moves to the uterus where it develops until calving 9 months later. On this video, you will view the events that the early embryo undergoes from fertilization until day 7.5 of development. This is a newly formed cow embryo. The number on the right-hand corner represents the time in hours relative to fertilization, when a single sperm cell penetrated the egg and shared its DNA with that of the egg to form a new organism, the single-cell embryo. A single-cell embryo is known as zygote, from the ancient Greek meaning to join. The embryo itself is surrounded by a coat of protein called the zona pellucida, which keeps it intact and protects it. Notice that there is a sperm cell in close contact to that zona. Let's watch the embryo as it develops in culture. Here, at 30.7 hours since fertilization, the embryo has divided into 2 cells. Each of the cells contains all the genetic material, the DNA, needed to form a calf. Each of the 2 cells are called blastomeres, which is the name for cells of the early embryo. Now, at 40.2 hours, the embryo has divided again into 4 cells. Note how active the material inside each cell is. These cells are alive. There are specific structures inside the cell important for its function, and they're actively moving. Inside each cell, molecules are being transported. New proteins are being synthesized, and fuel substances are being consumed to produce the energy for the embryo to develop. At 48 hours, this embryo has divided into 5 cells, and 30 minutes later into 6 cells. Notice that the cellular divisions are not always synchronous. It doesn't always double in number with each round of cell divisions because not every cell is divided at the same time. At this time, 91 hours after fertilization until 104 hours, this embryo contains from 9 to 16 cells. Important events are happening inside the embryo. It is at this time when the embryo's own genes are first turned on. Before this, the genes of the embryo are turned off, and the embryo rely on materials stored in the egg. From here on out, the embryo uses its own genes to control its development. Now, let's watch as the cells of the embryo continue to divide. Notice that with each division, each cell gets smaller, but the total number of cells in the embryo increases. As development progresses, it becomes more difficult to see the boundaries of individual cells. This is not an optical illusion, but occurs because the cells start to contract in on themselves, become compact. When the embryo has more than about 32 cells, we call it a morula. It resembles a mulberry. In this embryo, this event occurs at around 110 hours. At 128 hours, the cell starts to compact even more, and you can no longer see the individual cell boundaries. The embryo is a compact morula. Now, at 153 hours, see how the embryo has increased dramatically in size. It is increasing in size because the outer cells of the embryo are pumping water into the middle part of the embryo. The fluid-filled cavity is called the blastocele. The embryo now is called an early blastocyst. At hour 157.5, you can see the fully formed blastocyst, which contains around 100 to 200 cells. Now, for the first time, not all the cells of the embryo are functioning the same. The outer cells, the ones pumping water into the cell, are called the trophoblast cells and are fated to become the placenta. The inner cells are located in a clump called the inner cell mass. These cells are fated to become the fetus and eventually the calf. See how as the blastocyst continues to grow in volume, the zona pellucida starts to thin and the embryo continues to get bigger. At hour 160, the blastocyst has greatly enlarged and the embryo is called an expanded blastocyst. Observe that the embryo changes in volume. The blastocyst expands and then deflates, and the process is repeated. Eventually, the zona will thin to the point where the embryo is able to break out of the zona pellucida. You can see this occurring at hour 179. The process by which the embryo escapes from the zona pellucida is called hatching. By hour 185, the embryo has fully emerged from the zona, and it is now a hatched blastocyst. This is as far in development that the embryo will go in culture. If the embryo was in the cow, it would transform itself into the placenta and fetus and become a calf. This process that you just witnessed is very similar to what happens in other mammals, including humans. So, at one point, you looked a lot like this embryo, except more handsome or pretty. In the cattle industry, embryos are oftentimes produced in the lab to increase the number of offspring from genetically elite females. Embryos are produced in vitro, in other words, in the lab, and transferred at day 7 of development to a cow or heifer. Now, let's look at the video again and see the whole sequence of events without interruption. Once the egg gets fertilized, then it undergoes many rounds of cell division. By the blastocyst stage, it begins to develop into different cell types. The trophoblast cells will form the placenta, and the inner cell mass will form the fetus.
Thank you for watching this video, and I hope you are able to give you an idea of how the early embryo stages of development occurs.